They are related to food intake mainly, food intake, and subsequent to that there is the digestion and so visceral senses they are. Now this olfactory or smell sensation, it has its receptors. Receptors are in the olfactory epithelium. Olfactory epithelium. It harbors the receptors, smell receptors or olfactory receptors, epithelium. Epithelium. And here the peculiarity is that the receptors are neurons themselves. Neurons themselves. Olfactory receptor, olfactory epithelium. That is, it is situated at the roof of the nose. Two sides together, 5 centimeters square. It is relatively small than some other mammals like those. They are called macroscopic, where relatively high, where acuity, sufficient acuity, but is less than microscopic. And on its side, 2.5 centimeters square at the roof of the nose. That is on either side of the nasal septum. It contains the roof on the medial side and on the lateral side the superior turbinate. And this is the area. This is the area. And here epithelium is yellowish color, and they contain millions of neurons. Neurons are the receptors. And they traverse to the cribriform plate of the ethmoid bone. And these receptors, they have peripheral dendrites. And from this dendrite, they are the cilia that emerges. And this action, unmediated action, so cribriform plate of the ethmoid bone, and they will travel not single but in groups. And these are this called olfactory rod also, and these are the cilia, 10 to 12 cilia, and they are embedded in this fluid, secreted by the supporting cells, and to some extent some cells, bone cells that secrete this fluid. And this fluid is required so that they can capture the gaseous molecules. And the cilia, they bear these receptors. And they bear these receptors. Bear these receptors. They traverse through this in bundles, 20 bundles or so on each side, and they olfactory bulb. Then they have terminate into this type of structure. These are the cells itself, and it is the area where the nervous system is in close contact with the environment. These actions are unmediated action, and they traverse to the cribriform plate of the ethmoid bone, and they end in this part. This is the olfactory bulb also, and they are surrounded. They are not just bare; they bare, but they are surrounded by some non-myelinated, but there are some special types of, there is the cells similar to that of swan cell also. They are surrounded by this. There is some speciality regarding this. Detail you can have it in the anatomy test, Gray's anatomy also. Now in this way they form bundles and olfactory bulb, they terminate. And these cells in comparison to other neural elements, they are replaced. Within weeks, they are replaced by the differentiation of the basal cells. Olfactory epithelium contains these olfactory cells, that is the receptors, sustentacular cells or supporting cells, and the basal cells. And basal cells do replicate and differentiate and give rise to this type of receptor cells, and they are replaced. It is a unique situation, and it goes continue throughout life. It is going on. And these cells then they form the olfactory glomeruli, olfactory glomeruli, olfactory glomeruli, and the second order neurons, in this case mitral cell and tufted cells, mitral cells and tufted cells, and with which they form the glomeruli. They form the glomeruli. Yeah. Glomeruli. And glomeruli, from the glomeruli, then these fibers, second order neurons, mitral cell, axon cell, mitral cell, they traverse the olfactory tract. Olfactory tract. And they terminate into the olfactory cortex. And it is the only site where the sensory system, the thalamus is bypassed. Clear? In all other cases, in cutaneous sensation and other sensations that you have learned, that all go through thalamus. From thalamus, the fibers ultimately go to the respective cortical area. But it is different from this case. It enters directly into the cortex. It has connection to the thalamus, but that is secondary. Secondary. And these glomeruli, they collect good numbers of actions. Good numbers of actions. Collect. And their numbers are not on either side. That is 1800 number of, that is the glomeruli. And it is the mitral cell or tufted cell, and they spread in this glomeruli. And glomeruli is the site for the, that is the, it is second order neuron synapse, and it then enter into the appropriate area where the details of the smell sensation can be detected. Olfactory epithelium, we are discussing olfactory epithelium. Olfactory epithelium, that is situated at the roof of the nose by the either side of the septum and laterally by the superior turbinate. And they contain 10 to 20 million of receptors. And they are the neurons itself. And they have this peripheral process as the dendrite, and the relatively short process. And the terminal part is a relatively, it is a knob-like structure. It is called olfactory rod, from which cilia do project. And they are embedded into the fluid part, that is secreted by the either by the sustentacular cells or by cells just below the epithelium. And the cilia bear these appropriate receptors. And the cilia are non-myelinated and they form bundles. They are surrounded by some neuroglial, special neuroglial cells, and they enter into the olfactory bulb, and where they unite with the synapse with the second order neuron, mitral cell and tufted cells, and they then proceed in the olfactory tract, and they terminate in the olfactory cortex, principally situated in the uncus and parahippocampal gyrus grossly, and it is this place where thalamus is bypassed. In all sensory area or all sensations, thalamus is the obligatory station from which thalamic radiation bring it to sensory areas, and it is the place where it enters directly into the olfactory cortex. Now these millions, they terminate into glomeruli. Their numbers are not so numerous. On each side, 1800 of these glomeruli, where they terminate with the mitral cells and tufted cells. And this second order neuron, in this case, the axons of the mitral and tufted cells, and they proceed. And the olfactory tract, and they divide into three parts. Important is the lateral strap, intermediate, and the medial branch. And the lateral olfactory tract is then goes into and to this on the ipsilateral side, they terminate in the olfactory cortex in different areas. So that I am writing this because I can commit mistake. So I am writing this. Is this method of spread? Suppose we'll find the figure in reliable text. Suppose olfactory bulb, they are traveling. Mitral cell and tufted cell, and there is the olfactory epithelium, and they are tufted cells, and at different stations. At anterior olfactory nucleus, olfactory tubercle, then piriform cortex, amygdala, and then entorhinal cortex, contralateral olfactory bulb, and in this case it is the thalamus, orbitofrontal cortex, and also they traverse through this directly into the frontal cortex. Beside this, there is vomeronasal organ, and it ends into mitral cell, and then it is accessory olfactory area. In accessory olfactory area, and it traverses and it goes to the amygdala only. Amygdala. And from amygdala, hypothalamus. Amygdala to hypothalamus. And from this also this hippocampus. So this is the pattern. This is the pattern of supply. That is the olfactory epithelium. Millions of actions they do traverse. They are unmalinated and but they form groups. Suppose 20 bundles on either side and they are together are called the olfactory nerve. And they go into olfactory bulb on that side and they traverse in this way. And it is the anterior olfactory nucleus from which the second order neuron may arise or it will traverse through this. And anterior olfactory bulb from nucleus to contralateral olfactory bulb on the opposite side. It will traverse. Opposite side. এগুলো বই থেকে দেখে নেবে anterior olfactory nucleus and it contralateral olfactory bulb and then it is going through the other station olfactory tubercle by the intermediate tract and this piriform cortex, prepiriform or piriform cortex. That is smaller areas is close to that of the optic chiasma, lateral to that piriform cortex, and then amygdala at the anterior pole of the anatomically at the temporal lobe, close to that of the tail of the caudate nucleus. It has numerous connections. It has also contained other groups of nuclei. Detail you can find in Gray's Anatomy or any book. And then entorhinal cortex. That is this. It is mainly this uncus and parahippocampal gyrus. It is the area. Besides this, there are the vomeronasal organ. In humans, it is relatively small area at the anterior part of the nasal septum. It is chemically different and they have their receptors that terminate into this accessory olfactory bulb and from which mitral cells and they have their termination in the amygdala and then to the hypothalamus and they have respond to some chemicals that has pheromone function, that is the food intake and reproductive behavior that is regulated by this. In human beings it is relatively rudimentary but there is the existence. So this is this pattern of supply, and when you go through any reliable anatomy text, you will find much discussion. It is very difficult to remember everything in details. There are smaller areas that different names. Details of anatomy here you can have their information. Now their functions are different to some extent. Suppose from the thalamus orbitofrontal cortex and they are related to this recognition, not only identification, all the discrimination of the smells along with part of the frontal cortex and entorhinal cortex, principally the uncus, and it is related to hippocampus. Olfactory memory is related to this. Olfactory memory related to olfaction. It is principally done by the entorhinal cortex. Details of the different parts, then you have to read the anatomy text, Gray's Anatomy, relatively current edition, and they have discussed. And it is relatively complex but not intelligible, it requires labor. But physiology text, we do not require so much data. We have some basic concepts regarding this. And the number of glomeruli is much less than that of the numbers. But we can recognize different orders. More than 10,000 different orders we can recognize. And how it is possible? How it is possible? Recognition of different orders. Suppose 10,000 or even more than that, we can identify, discriminate different orders. And most of the orders, they are the mixture of these pure orders. They are not mixture of different orders. And how it is possible? For this type of structure, relatively anatomically simple structure, they has the power to recognize. And in this case, how it is possible? The nature has devised some method by which the simple structure can recognize them. And what are they? That is every receptor neuron. Every receptor neuron, they produce a particular receptor. Suppose if you divide these millions of 20 millions into 20 million neurons, 20 million divided by 1000, if you have get this type of groups that each produce a single type of primary odorant molecule, receptor for the odorant molecule. এটা বুঝতে পারা গেছে? And every action that enters, they divides into and they go into two glomeruli. That is, divergence in that case is not much. And this glomeruli, they are the site and they are placed two-dimensionally and their cerebral connection, orbitofrontal frontal cortex, or this entorhinal cortex, their olfactory cortex, and they have particular areas for their recognition. You have understood how it is possible? Suppose 10,000 primary orders, and if you divide 20 million by this 10,000, you will get 2,000. And if this 2,000 different types of neurons are there on two sides, and if each axon divides into two and they enter into two glomeruli, and glomerulus numbers are not more, and each group try to enter into their specific glomeruli. So if we consider that is the 1800 glomeruli, they are specific for particular order and their distribution pattern is also different. You can draw any figure and if you can divide into different groups, innumerable permutations and combinations is possible. Nature has utilized. So that the pattern of stimulation or the area of the stimulation of an olfactory bulb for particular odorant is different and it excites the different specific part of the olfactory cortex. And in this way it is possible to have the work of this simple organ. এটা বুঝতে পেরেছো তো? মানে particular type of, they will prefer some glomeruli and in this way they can have their information within the system. Molecular mechanism, whatever it may be, the receptors are different and relatively this type of genome is present. 1% of the total genetic information is for the odorant receptors, and it is more than that of the T lymphocyte receptors and the immunoglobulin molecules together. So a large part, 1% of the genetic information is devoted to the olfactory receptors. receptors. And in this way, it has been possible to detect different odors. When we are taking some natural odors, then they are not pure odors and they are combined, and then their different areas are simultaneously stimulated, and in this way we can get mixture of disorders. So the number do increase in reality, which we can, that is, it may be lakhs or so, that we can identify. Now, what is the basic mechanism? Receptors are different. But their other part remains same. Clear? The basic part is that these receptors are to some extent different, and they are, so that three-dimensional pattern is different, but their secondary parts, they are constant. Nature is much more intelligent, and it knows how to manage every case. So that it is possible by the help of these G proteins. G protein is called G-ol. Suppose it is cell membrane, and it is it unites with the particular odorant. And then there is the G proteins, alpha, beta, and gamma. G proteins, GTP binding proteins. When these molecules, odorant molecule, unite with this, normally it is in this position, and when this odorant molecule unites in this position, then what occurs? Then this alpha unit and they become separate, and alpha then relative to, there is the GTP, and beta gamma unit, they remain separate. It is this membrane, and it causes the activation of adenylyl cyclase from ATP to cyclic AMP is produced. And the cyclic AMP then binds with a receptor. Cyclic AMP, suppose it is the cyclic AMP molecule. Cyclic AMP molecule binds to this, and it becomes open, and then sodium ion and calcium ion, they do enter. This calcium ion then unite with another receptors, calcium, and it opens a chloride channel, so that chloride can enter. And when this entry of the chloride ion, then initially there is depolarization, and depolarization facilitated by the entry of the chloride ion, and in this way, when it is depolarized, there is liberation of the neurotransmitter glutamate from this, and they are acting upon, and when it is sufficient, then action potential will be generated. If the concentration is low, then generator potential is produced. And here there is the receptors at the neurons itself, and it is peculiar, they are also replaced. And whenever there is this type of events occur, then they become depolarized to such an extent, when the threshold limits cross, then generation of the action potential, and it will propagate. Now these receptor molecules, Now these receptor molecules, they are different, but these other mechanisms remains constant. Other mechanism remain constant. Other mechanism remains constant. Receptor molecules, they are 7 transmembrane domains and they are different. They are different, but this molecular mechanism after that is constant. And they then involve the different glomeruli, and these different glomeruli, they are activated according to the appropriate receptor. And in this way, there is a two-dimensional area of the olfactory bulb on either side, and there is also communication with that of the other side. Communication on side, so that the particular area of the cortex, they are produced. And in this connection, it is the power of adaptation. And relatively, it acts as the peripheral site. It has some effect on it becomes insensitive. And how it is possible? That at the peripheral site there are some changes in the receptors. Suppose in this process of, that is this molecular level, we consider that there is entry of calcium ion. This calcium ion, when this concentration rises, it unites with calcium calmodonyl complex, and it somehow lowers the affinity of cyclic AMP to this appropriate channels, appropriate cation channel. Is one mechanism. Or by some other mechanism, there is this calcium that enters, that increases the exchange of sodium with the calcium. So the calcium level do go level. Or there are some activation of some other compounds, then that can have their effects. And everything is not clear. You will find in different texts, they are expressed in different way. But the receptors becomes less sensitive. Threshold rises and they become less sensitive. Primarily with the entry of calcium calmodonyl complex, it either causes this cyclic AMP to unite with these appropriate receptors, or this calcium calmodonyl complex by means of activation of some enzymes causes the inhibition of the adenylate cyclase itself and produce the formation of the there is this cyclic AMP at the first level, or it increases the exchange of calcium sodium. This calcium is then pushed back to the outside. In this way, they have their effect. Adaptation is a property. And you have also learned how to test these nerves. In testing these nerves, you have to take the individual nostril. You have to close one side, and odorous substance that must be free from any pungent feelings. Now this olfactory epithelium, and they are close to that of the respiratory epithelium. And respiratory epithelium, they are supplied by the trigeminal nerve. And there are some compounds, elements or gases, they have double effects. They have effect upon the olfactory epithelium, and they also irritate these trigeminal nerve endings. And there are some feelings, relatively pain receptors, they are pungent feelings also. So when you have to test this olfactory nerve isolatedly, you have to take some substances, they will not stimulate this trigeminal nerve. There is the oil of clove, tincture of aspidida, and they are better. They will not stimulate. Ammonia is not. Oil of peppermint do have its effects. So low concentration can be taken. oil, cinnamon oil, a tincture of asparagus, they are ideal substances by means of which they can be tested. They can be tested. You ask the individual to close the eyes and close one nostril, and from a distance you bring this, and when at a distance he can smell, then you either speak or by raising the finger in this way that you can consider the distance. Similarly, on the other side you can test. You can test. In this way you can, that this side is affected. Initially you can test simply whether there is obvious obstruction in the, obvious obstruction. In that case it is, this less distance is there. So these are these different parts of this. There is the olfactory sensation. Olfactory sensation is combines with that of the sense of taste and other to have the flavor of the any food, flavor of any food. Now, as they are normally, there is a term that is called sniffing. When you are having smell at a distance, or from the nature, olfactory stimulation is always there, and they are always active to some extent. And in that case, when we are inhaling normally, small amount of that air that enters, into this area, enters the area, small amount. Because by means of diffusion or some convection current, epithelium, they have temperature more than that of the air entering. So there will be some convection current that is produced. That brings this. Into the air. But for better understanding, you have to sniff. Inhalation, at the same time there is the nostrils are relatively closed, so more air goes into sniffing. It is a automatic reflex response. If some new odor is present nearby, then there is sniffing, there is almost reflex. But you can also do have voluntary sniffing. Then in that case there is the septum, there is the lateral part of the nose will approach the septum. So more force will be there and more air will be going into the attic upper part, and they will be exposed. And it's called sniffing. And along with sniffing, then there is increase in the chance of this. Now this olfactory receptors, the olfaction is related to some reflexes also. Some reflexes. And these reflexes may be due to purely olfaction or due to the activation of the trigeminal nervous. And when there is the smell of a good food is coming, then there is salivation. Or when irritating substance is there, then there is sneezing. It is not due to olfaction, it is due to the involvement of this trigeminal divisions. There is sneezing, there is lacrimation. ফোড়ন দিলে কি হয়? ফোড়ন দিলে পরে আমাদের lacrimation হয়, sneezing হয়, lacrimation হয়. But the salivation, due to this purely olfactory salivation, they do occur. And it is through this route, there is this olfactory route, then even this, there is a reticular activating system can be stimulated. It can arouse them, individual, or make the individual alert of this situation, smell, particular smell, if it is harmful. So that in this way, and if strong, even respiratory inhibition may be there. And these are the functions of the nose. In that sense, it is not the olfaction really, but it is due to the effect of the trigeminal division of the nerve. Respiratory inhibition. And if smell is disagreeable, then there may be vomiting also in some individuals. Vomiting also in some individuals. So they are the related to reflexes and sniffing. Sniffing. Now, smell sensation, taste sensation is related to that of the smell. So, the foods that satisfies us in different components. Not only there is the taste itself, but the smell, taste, texture of the food, its temperature, they all do matter. All do matter. Yeah, all do matter, and they are termed together as the flavor. FLAVO. Now we can summarize our discussion in a few minutes. That is, the olfactory epithelium is situated at the roof of the nose, nasal septum, and roof and the neighboring part of the superior turbinate. And this epithelium, they have three types of cells. Olfactory epithelium, there is the neurons itself, they are the receptors, and sustentacular supporting cells, and the basal cells. And basal cells do proliferate, and they replace these cells in weeks. And the unmonitored actions of the olfactory cells, they enter through the cubiform plate of the ethmoid bone, and they form the bundles. And the bundles together, they are called the olfactory nerve. They end in the olfactory bulb, where there is the second order neurons, mitral cells and tufted cells, and they form the glomeruli. And second order neurons, they have their action, and they travel in olfactory tract, and then it divides into three parts: lateral, intermediate, and medial stria, and different parts of the olfactory cortex. From there is olfactory nucleus, anterior olfactory nucleus, they traverse to opposite side, and also a different part, which is olfactory tubercle, piriform cortex, amygdala, and entorhinal cortex. And there are some areas in the anterior part of nasal septum which is called vomeronasal septum, that is much more developed in case of rodents, and they have same G protein coupled receptors, and they are also united in the mitral cells in accessory olfactory bulb, and their actions terminate into the amygdala. From the amygdala to the hypothalamus, they have some pheromone action, and they are related to food intake, and also they have reproductive functions. Clear? Now this olfactory tract system is that it does not traverse to the thalamus like other sensory neurons, and they do enter into the thalamus by dorsal nucleus, and they have contact with the orbitofrontal cortex, anterior and lateral orbitofrontal cortex, and they also have some part of the frontal cortex also, besides activating this entorhinal cortex, which consists of this uncus and paraopicable gyrus. That is the entorhinal cortex. They are related to hippocampus and related to memory regarding this. And if olfactory epithelium, there is this primary neurons, they have their peripheral parts as there is the dendrite, terminal part has a knob, and from which cilia do appear, and the cilia are bathed by the fluid. The molecular mechanism of olfaction is that they harbor the different types of receptors, olfactory odorant receptors, and more than 10,000 primary odorant molecules are possible. And though there are millions of receptors, and these receptors, they are specialized for a particular odorant. Every receptor, they have this, and they enter into 1 or 2 glomeruli. So 1800 glomeruli on either side is sufficient. And if you consider, and they form a 2-dimensional map, and that is primarily responsible that we can discriminate 10,000 different odors. And they activate different areas. Orbitofrontal cortex and part of the frontal cortex, they are primarily responsible for this discrimination. Amygdaloid, there is complex that is connected to, besides this classical pathway, all the vomeronasal organs, they are related to hypothalamus, and they have some direction relating to sex and also this feeding behavior. And this threshold of some substances are very low. As for example, that is the methyl mercaptan, artificial mass. In methyl mercaptan, that is the substance that is responsible for the odor of the garlic. They can be recognized even at the 400 picogram ml, 400 picogram per liter. But it is much high for other substances like ethyl alcohol, ether also. We can discriminate different types, but the intensity discrimination is to some extent limited. Intensity discrimination, and it is relatively crude. So 30% change is required to have the differentiation. In comparison to vision, only 1% change in the stimulus intensity we can detect. And here 30% difference. In case of vision, in light intensity, if we consider millilambert or so, only 1% difference we can detect. But in this case, 30% difference in this case. It is similar to that of the taste. Another important factor is this is the adaptation, power of adaptation. It may be good smell or it may be disagreeable odor. We are become accustomed to after residing at that area for a long time. And the receptor, and this part is due to mainly to the effect on the receptors itself. Calcium ion do enter, and they form calcium calmodulin complex. And in calcium calmodulin complex inhibits the cyclic AMP to become attached to the receptors, cation receptors, or it causes this less formation of this by inhibiting calcium calmodulin complex. They do have some action of the protein, phosphorylate some other proteins, so that it causes inhibition of the adenylated cyclase. So cyclic AMP is less produced in the primary cells, or it can facilitate the exchange of calcium and sodium. So calcium will move out, and this process is retarded. Adaptation. Clear? And there are some terms such as anosmia, hyposmia also, and some diseases. Common cause is common cold. And with age, it is to some extent reduced. And some diseases like the fracture of the cribriform plate of the ethmoid bone, the trauma, or some tumors that can develop in the different tracts, these tracts, olfactory bulb also. And in that case, then they are the rares. Common is the common cold. And how they can be tested, we have learned in the practical class. I also discussed to some extent. I think it is all for today. I deliberately not started this taste sensation. In the next class we can discuss about the taste sensation. অথবা তোমাদের যদি কোনো কিছু অন্য কিছু আলোচনা করা থাকে তোমরা বলতে পারো, স্যার আপনি ওই পার্টটা একটু আলোচনা করে দেন, তাহলে পরে আমার জ্ঞান বুদ্ধি অনুযায়ী আমি তোমাদের সাহায্য করতে পারি। এভরিথিং ইজ রিটেন ইন দি বুক, বুঝতে পারছো? অজ্ঞ ব্যক্তির অপেক্ষা গ্রন্থের অধ্যেতা শ্রেষ্ঠ। তোমার যদি মনে হয় এবং যে ব্যক্তির জ্ঞান জন্মেছে সে গ্রন্থের থেকেও সে শ্রেষ্ঠত্ব অর্জন করতে পারে। ইট ইজ আওয়ার প্রাইমারি টিচিং। নাও কাজে তোমাদের যদি কোনো মনে থাকে যে কোনো কোনো জায়গায় নার্ভাস সিস্টেমের একটু আলোচনা করা দরকার, হ্যাঁ, আর আমাকে যদি মনে করো যে আমার মতো লোকে আলোচনা করার উপযুক্ত, তাহলে আমাকে বলতে পারো যে এই পার্টটা আপনি একটু আলোচনা করে দিন। এখনো তো একটা দুটো ক্লাস আছে, সেখানে আমি আলোচনা করে দিতে পারি। তোমরা সেটা চিন্তা করে বলবে আমাকে, তুমি একটু চিন্তা করে দেখো, তোমার যদি মনে হয় তাহলে পরে একটু, তারপর সেটাও আমাকে আগে জানিও, আমি তারপরে আলোচনা করে দেবো আমার জ্ঞান বুদ্ধি অনুযায়ী। তাহলে এর পরের দিন আমরা টেস্ট সেন্সেশন আলোচনা করব। এছাড়াও তোমরা যেটা বলবে বা কোনো কিছু অন্য কিছু আমি হয়তো স্বতঃস্ফূর্তভাবে তোমাদের বলতে পারি। আমার ইচ্ছা ছিল আজকে স্মেল টেস্ট দুটোই শেষ করা, কিন্তু সেটা করা গেল। থ্যাংক ইউ।